Well, welcome everybody to the Department of Microbiology at Oregon State University. I'm the head, Steve Giovanoni, and I'm here to tell you how delighted we are that you are interested in our educational program and how happy we are that we have a chance to speak to you today. This, of course, is a very unusual year. We're limited in what we can do, so we've come up with some new ideas for how to get to know you and how to introduce you to the department through a virtual approach. So you're going to meet lots of people today. You're going to see a lot of things. Feel free to ask questions of any of the students or faculty you encounter today. I know things are going to come up. You're going to want to ask questions later, so please feel free to go ahead and do that. I'm standing right now in front of a microscope. It's a very unusual and special microscope. It's actually a system that was designed by one of our own graduate students here a few years ago. The way this system works is cells, singles of microbial cells, are sorted on a microfluidic chip, and they're amplified to retrieve genomic sequences. This is one of the many exciting technologies and types of research you're going to learn about today. I hope when it's all over, you're as excited as we are. Thank you very much for being here with us.
Well, I think what OSU Microbiology does that is really at the top is it really integrates a variety of different microbiological approaches with data computing and bioinformatics, which is really where a lot of the field is going. So it's not like separate sections, it's all integrated into one. So one of the real strengths of the program is that integrated nature of the future of the field, rather than being sort of forced to look only back and not to see where everything's going.
They can expect to really acquire a lot of knowledge and skills in analyzing human microbiota in general, and microbiome in the environment, but also associated with human and animals. There is a lot of expertise in the department in analyzing big data sets, but also a lot in looking at what's happening at the cellular level.
We're very interdisciplinary. That is, we have students studying a variety of different types of microbiology, anywhere from food microbiology to marine microbial ecology to pathogenesis. So a very diverse department. In general, a lot of the students do collaborative work. For example, might work on some mechanism of disease, some pathogenesis, but might also do some genomics.
Trying to understand things at all of these levels, so biological, chemical, and physical level, to understand how all of these processes come into play and drive the microbial interactions that we're all probably investigating in our research. So it really comes down to kind of this interdisciplinary research that we bring into microbiology.
One thing that differentiates the Oregon State University Department of Microbiology from other related programs across the country and even around the world is the interdisciplinary and collaborative nature of the research that takes place on our program. The trainees receive exposure to a wide array of different types of research disciplines, techniques, concepts, and theories, and interact routinely with one another to resolve important and complex problems using these interdisciplinary approaches.
So I'm really interested in how the ocean works. And in many cases, that takes me to sort of far-flung places to figure that out. I have a significant amount of work that I do in Antarctica. Recently, we discovered a methane seep. It's the first active methane seep ever in the entire continent. And we've been trying to understand how the microbes there respond to an input of methane, which is a very potent greenhouse gas. And it's taken them years to start to begin able to sort of keep it out of the ocean, which keeps it out of the atmosphere. So understanding how the ocean fits into the atmosphere, fits into society is really a core component of my research. I also work in the deep sea on methane questions, and then shallow water on corals. And each one of those is trying to understand how microbiology interacts with animals, interacts with biogeochemistry, to understand how the ocean couples with society to a healthy planet.
So my lab, we focus on the viruses and bacteria associated with aquatic organisms, primarily threatened species. So we have a strong conservation focus in my lab. We study places like that, which are the tropical reefs of the Central Pacific or Florida. So we study coral microbiology, coral virology, but we also study fish, seagrasses, kelp, algae, and even marine mammal microbiology. So we're really interested in how bacteria and viruses might contribute to the health or the disease of these marine animals or plants.
In my lab, we try to change the microbiota in the mouse gut and try to see how that affects their behavior. But we also look on how specific bacteria will attach or not to sensory cell in the gut, and how does that impact the activity, for example, of the vagal nerve. So the vagal nerve goes from your gut to your brainstem. So those are direct connection of the gut microbiota with the brain. So we try to look into that in detail and analyze the microbiota. We also are developing, organoids, a new organ on a chip technology to really look into mechanism at the cellular level, try to understand how the microbiota interact with the hosts, more in terms of mechanism than behavior per se.
The CGRB is a crown jewel of the research infrastructure at Oregon State University, and students and other researchers in the Department of Microbiology routinely leverage the Center for Genome Research and Biocomputing for a variety of different applications. The first is the CGRB provides opportunities to help microbiologists generate data, especially but not necessarily exclusively for DNA sequencing-based investigations. And in particular, the CGRB houses Illumina platform high-throughput DNA sequencing machines, and they tend to be the go-to organization for producing next-generation sequence data. Second, the CGRB manages a very extensive, dare I say, world-class biocomputing cluster. The CGRB provides a series of important training, workshops, exposures, and opportunities for students in the Department of Microbiology, as well as elsewhere across campus. These include courses like crash courses in Python programming, how to use the Unix command line, R programming, so on and so forth. These training workshops also provide students with exposure to other trainees who are at similar points in their training arc in bioinformatics from across the university. They can form camaraderie, collaborations, and other types of beneficial interactions for training development.
So hi, I am Hannah. I'm the newest faculty member here. I'm a virologist. And so I'm going to take you on a tour of my lab, which is in progress of getting set up. So interviewing virtually is really weird. I did that last spring. So good luck, and I hope that I can show you around. Thanks. So we got, there's lots of space for people to work. So probably 2 people at this bench, a nice short bench here. So especially for that refrigerated centrifuge, if you're a short person like me, makes it a lot easier to change out your rotors. Then we can fit another normal working height bench here, so we can fit 4 people. I've got some biosafety cabinets because I work with pathogens.
So my research is looking at the mechanisms by which these bacterial pathogens cooperate with influenza virus in both initial acquisition of the pathogen and then also in dissemination of the pathogen to other sites like the lungs, where it can cause much more severe disease. And then also looking at host-to-host transmission and looking potentially into how does avian influenza cross the species barrier, and what impact does the avian microbial community have on that transmission event.
We go out to Hatfield Marine Station because we're interested in microbes that are found in the Oregon coast and in the estuaries. And Hatfield's really great because it's located in Yaquina Bay. And there's some really amazing field sites there. So there's a diversity of different type of ecosystems, from rocky reefs that you can study, looking at fish maybe in those areas. There's also mud flats that are really important for invertebrates that are found there, like snails and clams, etc. And then one of the things that my lab specifically studies is seagrasses, and looking at the seagrasses that are found just steps away from the Hatfield experimental labs, where we can go out and kind of go into the seagrass beds and collect seagrasses and then look at the microbes that are associated with those seagrasses to understand something about the seagrass health and the microbiome of these seagrasses. So there's amazing kind of field sites that are available. And then there are really wonderful lab facilities available at Hatfield. So there's a seawater pumping station that pumps millions of gallons of water into the facility every day. And so all these labs have access to fresh seawater. Some other really nice things about Hatfield is they have several ships available for research activities that go both near shore on day trips within the estuary and then far shore and extend it out into the coastal ocean and even into kind of past the shelf, I believe. And they should be getting 3 new regional class research vessels to do this type of research.
As an instructor in the department, I work with a lot of graduate students who are working as graduate teaching assistants, whereas part of their grad student responsibilities, they're teaching students, which can involve everything from running laboratories, helping students learn the fundamentals of microbiology, learning how to deliver lectures, to grade fairly, to support students as they grapple with these hard scientific concepts. I also get to interact with graduate students on committees that I'm a part of, such as the Core Values Committee within the Microbiology Department. And we're aiming to provide support and to address concerns and to improve the culture of our department.
I think it's important for students to understand that the faculty take trainee success very seriously. We invest our time, energy, and resources into promoting the professional development of our trainees. We want to see them succeed.
So thank you all for joining us today on this virtual tour of the Department of Microbiology. We certainly enjoyed getting to know all of you, and we hope you enjoyed finding out about us, and we look forward to further conversations with you in the future.